Hi, good morning, everyone. So today we're going to have this Outstanding Achievement Award speech. And it's my great honor to introduce my long-term colleague and a friend, Jim Bennett, who is the winner of 2018 American Society Gene and Cell Therapy Outstanding Achievement Award. So first I would like to introduce Dr. Jim Bennett. Dr. Jim Bennett is F.M. Kirby Professor of Ophthalmology, Professor of Cell and Development Biology, and co-director of the Center for Advanced Retina and Ocular Therapy at the University of Pennsylvania. Actually, I have known Dr. Bennett since my time at Penn. I clearly remember 1999, and I'm honored to co-author with Dr. Jim Bennett for the first paper in PNS describing how we can use AAV to transduce photoreceptor cells in primate eyes. So since then, actually, Dr. Jim Bennett spent 26 years at the University of Pennsylvania, her lab, trying to develop gene therapeutics for retina and ocular neovascular diseases. Dr. Bennett's research started with trying to elucidate the mechanism of pathogenesis and retina differentiation and identify potential therapeutic targets and develop therapeutics for those diseases with gene therapy as a major platform. Dr. Jim Bennett, her lab has really developed a model from bench to bedside. And yesterday, Dr. Kathy Hai presented the first US FDA-approved drug called Luxturna. Actually, Dr. Jim Bennett was the one really started from the basic research, proof of concept, translational experiment in non-human primates, and eventually in dogs, and then moved that to clinic. And with Dr. Kathy Hai's collaboration, they were able to finally develop this as the first gene therapy drug, in vivo gene therapy drug in US, and now become commercialized. So I think at this moment, I would like to represent American Society of Gene and Therapy and give this award, Outstanding Achievement Award, to Dr. Jim Bennett. Now I would like to present Dr. Jim Bennett for this Outstanding Achievement Award speech.
Thank you so much, Guanping, and all of you. I am so humbled to be the recipient of this award, and I only accept it as a representative of my extraordinary team. I am so grateful to all the people behind this, the committees, ASGCT, of which I've been a member since its inception. It's my favorite society. All of you, and especially the patients who so bravely and generously volunteered their time and participated in our studies. It's been an extraordinary experience for me and all of my team. So I'd like to start with this slide, which you see here, to illustrate how wonderful my experiences have been with my colleagues. Shown here is a photo of my lab in 2001, shortly after Lancelot's paper, which he's reading, was published. Lancelot was born blind, and he had gene therapy and began to see. And I came into the lab early one morning in December, and everybody was gone. I couldn't figure out where everybody was. They came back and handed me this photograph. They had all gone to the facility where our dogs used to live, which was 40 minutes away, and posed with this picture. And it posed with this journal article. And later on, I was inspired by their photograph and did a little photoshopping and submitted this to Nature Genetics, where Lancelot was awarded Mutant of the Month. That's what that picture is. So I'd like to start by disclosing my conflicts, and I'm also disclosing conflicts of my husband, Albert McGuire, who has worked with me the whole way on this project. We co-authored a patent early after we saw that the effects in the Briard dogs, so that in terms of rescue of their vision with gene therapy, and we immediately waived any potential financial gain. But that patent belongs to Penn, also Cornell and University of Florida, and it's been licensed to Spark Therapeutics. I also have the other disclosures to mention below. So this has been an extraordinary experience, as I mentioned, with extraordinary people. And before I say anything, I just want to show some photographs of the teams of people who have been involved in the Luxturna Veretagene studies. Shown on the top is the view of the whole team immediately the evening after the injection of the very first patient, and I'll tell you about that shortly. This includes our Italian colleagues who came specially for the injection of the first subject, who happened to be Italian. And then in the middle photograph is the team after the youngest child in our study received his injection, and he's shown forefront in the center. His name is Corey, and you'll hear more about him shortly. And that's the whole team that was involved there. It was a lean machine in those days. And then below, more recently, is my laboratory team at Center for Advanced Retinal Ocular Therapeutics, which we call CARET. So where did it all begin for me? In 1985, my husband and I were medical students, and we completed our senior project, which was the birth of our first child. And in the glow of happiness after that, my husband, Albert McGuire, said to me, "Gene, we did so well with this gene transfer project. Do you think we could transfer genes to the retina?" And the reason why he asked that is he knew that before I went to medical school, I did a postdoctoral fellowship with French Anderson, shown on the left. That was before French had actually run a clinical trial, but he was working in the laboratory to generate the reagents and prepare for this and see patients with the disease, which was later treated in 1990 in the first approved gene therapy study. And I was so excited about gene therapy and the promise of gene therapy that I asked him, "How can I do what you're doing?" And he said, "Simple. Go to medical school, go to Harvard Medical School, learn about the diseases, and then go back to the lab and start working on them." Well, I was fortunate enough to be able to do that. And then if we consider how Albert got into this, as a medical student, he had decided to specialize in the retina. He became a retinal surgeon, but as a medical student, he worked with Elliot Burson, who ran the first lab that was designated to study human retinal degenerations and characterize them. This was a lab funded by people, including Gordon Gund, who developed retinitis pigmentosa. And 40 years ago, instead of giving up, he decided he was going to set up a foundation to support research in this area. And so we were both interested in this from these different perspectives. But what I didn't tell Albert was, well, yeah, let's do that. What I didn't tell him is what we didn't have. We didn't know any genes at that point in time. The first genes involved in retinal degeneration were discovered in 1989, 1990. We had no animal models characterized, no natural history data, no vectors, no surgical methods to deliver genes to the retina, no outcome measures. We really did not have very much to do, but we were very naive, and that was actually a blessing. And as we continued our training, we wrote a little pilot grant to the institute where Al was doing his retinal surgery fellowship, which was funded for the tune of close to $6,000. which allowed us to get some pilot data. We did some non-viral gene transfer, developed the surgical techniques to deliver genes to the retina of a rabbit. And you can see here the lac-Z expression in this rabbit retina. It was transient, but it worked. And this was leveraged then for a career development award. The first proposal I wrote did not get funded for in vivo transfer, but they funded me for a different study which actually led to gene therapy approaches. And this was from the RP Foundation Fighting Blindness, now known as Foundation Fighting Blindness, again supported by Gordon Gund and Luli Gund, shown in the photograph. And I am so excited that ASGCT will be sponsoring many career development awards in the future to help people who are early in their careers and want to try things like this. Then we moved to University of Pennsylvania in 1992, and I was thrilled to hear that my previous companion in the medical school, in the wards at Massachusetts General Hospital, was being recruited to University of Pennsylvania to run what was called the Institute for Human Gene Therapy, and that is, of course, Jim Wilson. We worked together. He was an intern when I was a medical student, but I had no idea he was interested in gene therapy. And so that was news to me, and we didn't discuss that at all. He didn't know I was interested in either. But when he set up this institute, he developed all sorts of facilities which became very useful for developing gene therapy for the eye. And so we did studies with adenovirus and multiple different vectors, as shown here, testing out the various toolkit. And Guangping referred to one study, one of our first large animal studies involving AAV with a monkey, and I'm totally grateful to him and all of the other individuals shown in this slide here. The top ones were involved with either Jim Wilson or the Institute for Human Gene Therapy, and then later on, as we started working with CHOP and the RP65 studies, Shang-Zhen and Fraser. It was extraordinary. So then the next problem, the genes. As I mentioned, no genes were around when Albert asked me that question in 1985. However, they started to be discovered. And at this point in time, there are more than 260 different genes which, if mutated, can cause blindness. The RP65 gene was first discovered in humans as a cause of disease in 1997. And that made it a candidate for looking for causes of disease in animal models. The dog disease was identified as RP65 shortly thereafter. And lo and behold, we had some dogs. We had 3 dogs, to be exact, at University of Pennsylvania in a facility that was then being managed by Greg Ackland. So what is this disease? 2 mutations in RP65 cause very early onset retinal disease. Children are born with abnormal vision for multiple aspects, which we'll talk about, and there's a progressive degeneration, so they lose whatever vision they have ultimately. And they often have abnormal eye movements, as shown in this image here with the dog and with the human above the dog. RP65 is a gene expressed in the retinal pigment epithelium, which helps mediate the retinoid cycle. And so without it, 11-cis retinal cannot be provided to photoreceptors and they can't see. And Kathy Hyde described this really eloquently yesterday. So we went ahead, Greg Ackland, Al McGuire, and I, and injected these 3 dogs. And a couple weeks later, the animal caretakers called me and said, guess what? Lancelot's seeing. He's watching us walk around the facility. His head is following us. His behavior has changed. His whole demeanor, he's become very active instead of crunched in the corner because he's scared. And doctors Alleman, Sedacian, and Jacobson measured electroretinograms, which I'll show in a minute. Shown in the image is Dr. Alleman. And lo and behold, they were extraordinary. And this led to a marvelous career that Lancelot led for the next 10 years of his life. He attended various meetings in Congress, at least 3 of them, was a poster child for gene therapy, and led a very happy life with his wife and sister, Guinevere. and his some 60 offspring who then participated in our studies. And here is one that participated in the study. Before injection, he walks around banging into things because he can't see them. And afterwards, he walks around wagging his tail, not bumping into anything, and clearly very different behavior. And below that, you see, again, the electroretinograms, which I referred to. On the far left are wild type is a wild type retina, and you can see the characteristic waveform with the amplitudes at the increasing exposure to light. Before treatment, Lancelot had barely a blip, even at the highest light intensity. And then afterwards, robust electroretinograms, which persisted for the rest of his life. And also pupillary light reflexes. So in the untreated eye, if you shine light at it, it does not constrict. Normally, eyes do constrict with light, as seen here in this eye that was treated with gene therapy. And we looked at multiple other things, including the fact that younger animals responded better than older ones. But then, as Kathy High described yesterday, we were our funding dried up and things became very, very difficult because of the problems that were going on in the gene therapy field. And our plans for moving forward with a clinical trial with our previous collaborators were dashed because they decided to bail from University of Pennsylvania and move the studies to another institution. But then one day, Kathy High walked into my office and she described to you yesterday how she had had the idea of taking advantage of the failure of some of these companies by recruiting this specialist to her center at Children's Hospital of Philadelphia, known as CHOP. And she said, Jean, how would you like to run a clinical trial at CHOP? It took me like a millionth of a millisecond to say yes. And we were off to the races. And as Kathy described, one of the first steps, a very important step, was to generate the proviral plasmid, which would give maximal expression in the AAVs, which were generated. That was done by Jeanette Bennett in my lab. And there were various other tricks to make sure that there were minimal empty capsids, which was accomplished by Fraser Wright at CHOP. And we looked at studies in AAV dosing and found, which had not really been described widely before, that AAV, particularly at lower concentrations, can stick to inert surfaces. So you have to be very careful to be sure you're giving the dose you think you're giving. We took care of that by adding some surfactant to the brew, and now everybody else is doing that too. And then we realized there was no path for pediatric gene therapy drug development because people had not carried out gene therapy for children with non-lethal diseases before. So we had to learn about the drug development principles and the legislation and policy dealing with this, and then argue this case at the NIH Recombinant DNA Advisory Committee, the RAC, which we did one long day in December 2015. And we were grilled, and right at the very end, one of the individuals said, well, you know what? My uncle was blind. He navigated the streets of New York City. He played saxophone. He was a millionaire. It was fine. And we thought we had lost it right then. But then David and Betsy Brent, who are heavily involved with Foundation Fighting Blindness, stood up, as did another family with this condition, and described the difficulty they go through every day with a child with Leber's congenital amaurosis, including the fact that it takes 12 people to get their child through one day of school. And the response was, from the RAC was a unanimous approval. So we made a big difference there, not only for our study, but then the subsequent studies were also able to enroll children. So we were finally ready, but there were no available US subjects. Real problem. None of them had been genotyped or phenotyped, at least the ones who were available to us. So we were saved by our Italian colleagues, shown here, Alberto Riccio, Enrico Serace, and Francesco Simonelli. Francesco Simonelli runs a clinic for retinal degenerations in Naples, Italy. And she had wisely, thoughtfully characterized genetically and disease-wise all of her patients and had a whole list of patients with RP65 disease. And so our first subjects were Italian. We set up a site in Naples where they were screened at baseline, sent to CHOP, they were screened again, had surgery, and we did dual follow-up visits, so that added robust nature to the data. And I'd also like to point out the clinical coordinators and the other individuals there, which I'll get to. So the injection, this is how the injection evolved ultimately, and this was carried out by Albert McGuire. You see in the side of the picture, it involves introduction of a cannula that's about the width of an eyelash. So it makes a tiny little incision into the retina and delivers the material directly to the region where there are cells ready to benefit or respond to the gene therapy. And this localized retinal detachment flattens within a matter of hours, exposing only those cells to the reagent. So we began the phase 1 study, which was a dose escalation study, in October 2007. There were 2 other contemporaneous studies, one which had already been started by Robin Ali, and another which was about to start at University of Florida / University of Pennsylvania. And shown in these images are one of the initial tests we gave in an exploratory measure, the mobility test. And the subject at baseline stumbles through the course, bumping into things, feeling with his feet, but afterwards walking slowly but accurately through the course. Well, this was the initial test. The initial test was made of scraps of material I had in my basement, tiles, pieces of foam, things which were soft so that people couldn't stumble over it. And it went through a number of configurations helped by numerous people, and then further by Dan Chung, as I'll get to, for the phase 3 study. But shown here are the results of the youngest child in the study, Corey Haas. After his injection in one eye, one eye is patched and the other eye is not, so he's using his uninjected eye at the left, and he bumps into things immediately and can't find his way through the course, whereas with the other eye, and I don't know if well, we'll skip that, but with the other eye, he went right through the course and had no problem. We had another exploratory measure, which I loved, pupillary light reflex, which showed that there was a response in the injected eye, but not the uninjected eye. And there you see the waveform, the pupil constrict in the injected eye only, which in this case is the right eye. But it was also the bane of my existence because these individuals had nystagmus, and so I had to spend so many hours measuring the diameters of the pupils and graphing them out. There were 3,000 images per visit. And I was kind of glad when the pupillometer broke down and we couldn't carry this out anymore. So I'd just like to illustrate the impact that this treatment had on Corey Haas in particular and other children. Here he is walking through the hospital 2 days before his injection, using his blind cane and holding his parents' hand because he could not see his way to walk independently. And then within a year and a half after the injection, he was doing things that are clearly visual, riding his bike, hammering, playing board games, playing sports, basically living the life of a normal 9-year-old boy. And I keep thinking of him and the other children at this locked-in age. But Corey, fast forward 10 years later, is now 18 years old. This is him at his prom last month. And he did not need any help picking up his date. He had a great old time. And the week before, he had actually gone turkey hunting and caught a turkey. Well, you would argue that that requires some visual help. So what the subject said is various things that you can read here, which have changed their lives. They seem little, maybe, if you have really good vision, seeing stars and that they're not star-shaped. But universally, all of them asked when they could have their second eye injected. And this is something that Kathy spoke about yesterday, that we were also told by the FDA that we had to think about a plan of injecting the second eye, because if this gets approved, surgeons are going to inject both eyes. So the concern was, is it possible to improve visual function further by injecting the second eye? And of course, the chief concern was immune response, which could limit further benefit in the second eye, or perhaps even make the outcome worse for the initially injected eye. And there are very few success stories of re-administration of gene therapy. So we went back to our trusty animals. But the problem is we didn't have any more animals. We couldn't get them anywhere. But we had a solution, and that was thanks to Dr. Kristina Narstrom from Sweden, who was one of the first people who characterized this disease. She, in exchange for a cash donation that I gave to her lab, gave me some affected RP65 dogs that I picked up in Missouri. And then I had a new training plan. I had to learn all about dog breeding, AI, it's not just artificial intelligence, artificial insemination, whelping, puppy care. When the puppies were born, I had to go bottle feed them every 3 hours. That was a challenge, but it was also incredibly rewarding. Shown here is Corey with one of the dogs, which is now living at our house. We'll get to that. But the re-administration study used both monkeys and affected dogs, unaffected monkeys, designed to administer a high dose of AAV to one eye, and then when the immune response would be maximal, go to the second eye and administer it. And we evaluated a number of different responses, including immune responses, and shown below is Federico Mangazzi, who was heavily involved in these studies. And we had to carry our equipment back and forth to the various facilities. Shown there is Al, walking the microscope down Civic Center Boulevard, all sorts of challenges, but we got it done. And shown here are the results of Venus, representative of the others, who is clearly visual, avoiding all the obstacles in this course, as did her son Mercury. And shown below are Mercury and Venus, who after their studies were finished, we adopted them, and they are now living at our house, having a grand old time. We found some interesting other findings in these studies and related studies. We looked at sera and intraocular fluid derived from patients who were undergoing other procedures, and found that even in patients who had very high neutralizing antibodies in their serum, they had barely any neutralizing antibodies within their eye, within the anterior chamber fluid or vitreous. And that also, we also found the same thing in our dogs, and that assured us, or gave us some more confidence in terms of the immune nature of the eye, and allowed us to drop an exclusion criteria that we had initially in our phase 1 trial, which eliminated people from participating who had high titer antibodies in their serum. We also found that AAV2 re-administration did not result in harmful T-cell responses. So what happened with re-administration? Although we were assured by our data, we still proceeded cautiously because, as Dr. Hai pointed out, you don't always see the same things in humans as you've seen in animals. And shown here is the first subject who had her second eye injected, going through the baseline mobility course and stumbling, but afterwards doing a great job. This subject and the first 3 subjects stayed here for 3 months. Since she was from Italy and she didn't have family to come with her, she stayed at our house, which was an amazing experience as well. I wish I'd learned more Italian, but I did enjoy things like having her all of a sudden look in the mirror and go, "Mamma mia!" She saw her face for the first time. Visual fields also changed, and let me explain why that's important. Most of us, if we have wide fields, we can look at something like an elephant and see a whole elephant. People with tunnel vision, or very, very narrow visual fields, look at an elephant and they see basically a piece of the trunk. It's very hard to figure out what that animal is. And so the effects on visual fields were important too, shown if you just look at the top row there, results from one patient at baseline. There's a teeny little visual field shown in blue there, and after injection, the visual field has expanded and stays pretty stable over time as it's followed. Remember, this again is re-administration, so it was effective. And Manzar Ashtari looked at a lot of different aspects of the visual function in the cortex after gene therapy, which I don't have time to go to, but just for illustration, shown here are the results of administration to one subject, where you can see at baseline there's no response of the visual cortex when the person is shown images in the goggles, but afterwards you can see robust responses. So we found out information also about the wiring of the brain and the fact that it's intact and can be reactivated if the retina is rescued. And all of this required meticulous bookkeeping, and shown again is Jen Wellman, who was responsible for this and responsible for keeping us all out of trouble as well, which Albert really appreciated, being the PI of the study. So what about the outcome measure? The FDA had told us that we need a clinically meaningful result, and the problem is there's only one test which had been approved, and that's reading the eye chart. The problem is, in previous studies, the problem is many of these people couldn't even see an eye chart. So we were fortunate to have been able to look at all of our exploratory measures, and we chose the mobility test to further develop, as Kathy described, and that was done by Dan Chung, who was in my lab at the time and has since gone to Spark. And ran the clinical trial design, which Kathy High described the other day, which involves intervention to both eyes following for various periods of time, and then a control group that is then undergoes evaluations, but then crosses over to receive intervention after 1 year. So as we were getting ready for the phase 3, as Kathy described yesterday, we were thinking, how are we going to pay for this? CHOP was very generous for the phase 1 and follow-on studies, but phase 3 is a different order of magnitude. And we looked at various options, and shown here is the beginning of Spark, which it's a memorable day for Bev Davidson, Kathy, Jeff Marazzo, and I, and the others who founded Spark. And then soon thereafter, Spark was formed, and you can see Jen in the hard hat. And so Kathy also described the safety data yesterday, which I won't elaborate into. It was actually excellent safety profile, no serious adverse events due to the drug. And the results are depicted here in this mobility test, which had been standardized as described yesterday, and shown it is at baseline, the person having a great deal of trouble navigating the test, using her feet to feel things and stumbling into things, whereas a year after intervention, she found the course and found the end of the course in record time without hitting any obstacles. And there's another set of tests here, which I will probably whiz through, because you have the idea now that these people really were successful at being able to navigate this course. So here's the graph showing the responses of the mobility test of the people who received the intervention, and then those who were assigned to the control group and then received the intervention of the following year. And I want you to remember the shape of this graph, and now I'm going to whiz through to the endpoint for looking at light sensitivity, which is a totally different test, but you notice that the shape of this graph is very, very similar to what we saw in the mobility test, with rapid response in the intervention group, none in the control until they went over to the intervention group. And then in a third test even, in testing the visual field expansion, which is one we weren't sure we were going to hit, you can see the same sort of pattern, rapid expansion of the visual field, which is maintained, and then the control group went through the same thing a year later. We also had an exploratory measure using a visual function questionnaire. The National Eye Institute has a questionnaire that's used for many clinical trials, which asks questions which are irrelevant to a pediatric population, such as how much difficulty do you have driving in dark in difficult conditions, such as when it's snowing or in the dark, or how much difficulty do you have shaving or putting on makeup? Well, you ask a 4-year-old child about that, it's clearly irrelevant. And so we designed our own questionnaire to better reflect the problems these individuals have. So for example, if somebody accidentally leaves a cabinet door or a locker door or some other movable object in a new position, are you likely to bump into it? Or when it's light outside, do you need to kick your feet or grope with your hands to help find your way? And we graded this on a numeric scale as shown here. And what was found also in phase 3 clinical trial is that this questionnaire, oops, I just moved too quick. This questionnaire also showed that there was improvement in the individual's perception of their quality of life, but no significant improvement in the control group. So, how does improvement affect an individual? And I'll give you one specific subject. Shown here is Christian Gardino. He's quite a ham, as you can tell. This is right before he was going to have his second eye surgery, and he thought he'd fool Dr. McGuire by putting a handkerchief over his face. But you can see from the data shown in the slide that Christian, after the surgery, had a dramatic improvement in light sensitivity, both in chromatic light and in white light. And he also improved in his visual acuity, at least in one eye, quite significantly, more than 15 letters on an eye chart. He also improved in his visual fields. Shown here is his baseline and then post-treatment after in one eye, and here graphically. These fields are not perfect. Shown at this dotted line is what we'd expect a normal-sighted person to have using that same test, but it's pretty darn good. And this has allowed Christian to excel in his career.
Thank you.
What's your name?
My name is Christian Guardino.
And Christian, how old are you?
I'm 16 years old.
Okay. Tell me who you're here with, Christian.
My mom's here with me.
Okay. What do your school friends say about you coming on the show?
I only told like a few people, like my very best friend.
Your very best.
So Christian did not learn how to sing from gene therapy. He sang before, but when he'd go on stage,
I've been singing for as long as I can remember.
he would not walk around. He was scared. He had to be led up on stage. And he could not see people's faces, could not see where he was going. And there's quite a difference now.
Okay, Christian, best of luck.
Thank you.
Note how he's walking around below. And you can see he won the, whatever it's called, the golden parachute. So anyway, this is on YouTube, and you can look at it if you're interested further. So after the FDA advisory committee meeting last October, we were thrilled at the unanimous recommendation that this be approved. And we thank the FDA advisory committee members for their hard work in this. And then we were doubly thrilled to find out that this was approved as a drug in December of 2017. And just a few weeks ago, we were especially thrilled to hear that the drug had been administered as a prescription drug at 3 separate institutions near simultaneously, at Mass Eye and Ear, at University of Miami, and at Children's Hospital of Los Angeles. And we've since heard that these individuals have done well, and we don't get tired about hearing these stories about what they can do now. It's so exciting. So going forward, how can we speed up the process of developing AAV-based gene therapy treatments? There's so many other diseases. Remember, I told you there are more than 260 different genes that can cause a disease. And for many of these, we don't have animal models, or the animal models are completely irrelevant. So we tested a new approach by looking at induced pluripotent stem cells, iPS cells, and we used choroideremia as a target. So the idea is you take these cells, put them in a dish, dedifferentiate them, and then differentiate them to a retinal phenotype, shown here as RPE cells. test your intervention in the dish and see whether you see efficacy. And in this case of choroideremia, we looked at prenylation assays and we looked at protein trafficking. And then if you see that it's working, then do safety studies in normal-sighted animals and then go forward to start a clinical trial. Well, we carried out this, this whole process with choroideremia thanks to Spark Therapeutics, which is a sponsor of a clinical trial, which is now at the point where individuals are reaching their 3-year time point. And this trial is co-PI'd by Tomas Alleman. And we're very excited not only about the possibility of gene therapy for choroideremia, and there are other trials that are going on which are reporting very exciting results, but also for extrapolating this pathway to other diseases so that we can move quicker to get people interventions while they still have cells that can be rescued. In the process, Albert McGuire, Callie Stassi, and I have developed a subretinal training and procedure package based on all of our experience. This is through support through Novartis, and this is meant to be a guideline for people who want to get into this field and they don't know what they need. And we will make this available through our site, and also the Foundation Fighting Blindness will make it available on their website. What we don't want this to be is there was an article in the New York Times the other day about people being able to do cloning of harmful viruses at home. We don't expect you to be able to do gene therapy or subretinal injections at home. So in summary, gene therapy for the retina is alive and well, where there's 1 approved drug and 2 additional targets are in phase 3 trials. There are a number of people now enrolled in trials, more than 700, and we think that number is going to swell by the end of this year. The majority of these studies use AAV, and there are trial sites all over the globe now. And so we are very excited about what has happened with Voretigene/Luxturna. It took 17 and a half years from proof of concept to get FDA approval. Which Kathy High pointed out is competitive with the timeline for drugs which we now consider as standard of care. There were a lot of firsts along the way, including the first pediatric gene therapy for non-lethal disease, studies of adherence of AAV, randomized multi-center controlled phase 3 study, contralateral eye re-administration, functional vision outcomes, and an IND package based on data from iPS cells. And we hope that this now creates a path forward for genetic treatments for other devastating diseases. Now let me just end on one more point, which is timely. I mentioned French Anderson's role in my career in the beginning of my talk. Well, he called me. He is, as many of you know, he has been out of the land of the free for the past 14 years. He spent 12 years in prison. He was just released yesterday with abundant evidence of his innocence, and he called me the minute he got into his house. And one thing he said is that he hopes to learn to pick up all the progress in gene therapy and get back to it at some point soon. So I hope you'll welcome him back to ASGCT. So we're thankful to so many different individuals, and particularly our subjects and our families, and shown in that video are Venus and Mercury, who are clearly visual after 10 years after their gene therapy. Thank you very much.
So, hello. Thank you very much, Dr. Jim Bennett, for your great speech and for sharing your personal and professional journey in developing gene therapeutics for eyes. So from her talk, I'm sure that everyone in the audience, just like me, now get a clear picture of Dr. Jim Bennett. She's not only a giant, I used my colleague Luke Vandenberg's word used yesterday, but also a pioneer and outstanding researcher in gene therapy. And she is also very humble colleagues, and who always give the credit of her tremendous outstanding achievement to her mentors and to her colleagues, trainees, patients, and family. So now I would like to open the lecture hall for questions to Dr. Bennett. Okay, if we do not have further questions, let's give another round of applause to Dr. Bennett. Thank you very much.